Sick pod method is commonly used for the varietal evaluation against soil-borne pathogens under the greenhouse condition. Sick pod carry the field soil, which is heavily inoculated with the soil-borne pathogen in comparison to the control part which carry the field soil, but that soil is already sterilized and free of the pathogen. This picture will help us to understand the procedure involved in the development of sick plot. Initially, we need a fungal culture growing on the solid medium. That culture must be young, and we have to remove the disc of this fungal culture under the contamination free environment such as in the laminar flow, and then put the disc into a broth medium or the liquid medium so that the fungal culture may multiply, but we have to put this flask into a incubator shaker to facilitate the multiplication of the fungi. After that, once it is well grown in the broth culture, we have to mix the content of the broth culture with the sterilized field soil. And once the mixing is done, we put the content into the pot. And now this part is the sick pot. However, for the control part, we just add the sterilized field soil without any kind of the pathogen. In order to test whether our pathogen is well established into the sick pod, we have to add a few seeds into the sick pod of the most susceptible known variety. And if the plant die after the few days, it means that the pathogen is now very well established inside the pot, which we designated as the sick pot. Now, if we want to test the performance of our variety, we need to put the seeds of the test line into the sick pot as well as the control pot along the known susceptible variety which act as a positive control. And we have to leave these pots under the greenhouse condition for to observe the appearance of the disease. The possible outcomes of this experiment or the practical is the plants growing on the control pot, including the known susceptible variety, must be remain healthy, while the plants of different varieties growing on the sick pod may vary in response to the inoculation from susceptibility to resistance. However, plants of known susceptible variety must show disease symptoms, and the experiment or the practical needed to be replicated.